In this short video clip, I'm going to give you a preview of one of the organisms you will see in our lab. This is Volvox. Volvox is an example of green algae. It's a chlorophyte. You may be noticing very quickly that these are in no way green. They have been stained, and they are dead. But when you see them live in the lab, they will of course be beautifully green, and I bet you can guess which organelle is responsible for that. Anyway, Volvox, about 24 species. They like to organize themselves into spherical or oval hollow colonies, and these colonies are made of individual Volvox cells, each of which has 2 flagella. And 500 to 60,000 of these individuals will organize themselves. They're linked together by little strands of cytoplasm, which you will recall from our cell unit. And they reproduce both asexually when the environment is all calm and good and predictable, and asexually when life is riskier and you need more variety. So you will notice that almost all of these I'm going to show you have daughter colonies inside of them. A term that's used for those daughter colonies is gonads, because many of these species come in male and female varieties. They can divide asexually, but as I said, when times are tough, they can go out and find a date. So when the daughter colonies have undergone a whole bunch of cell divisions, they do this along the equator, along the middle of the big colony, and so when they're ready to separate from mom and go out into the world and go to trade school and college and that kind of thing, they find themselves in a difficult situation, which is that their flagella are on the inside. And like small children on a kicking board going across the swimming pool, they need to have their little leggies in the right part if they're going to row. So if you were so lucky in lab, when you see the living ones of these, to notice that one of the daughter cells is turning itself inside out, that's what I call Flippy Flagella, and they're doing that so that they can position their flagella on the outside so they can get out and row, so they can get out and kick, so that the whole colony can move, exhibiting a behavior you know is called phototaxis. So one structural thing that goes with this function is that they have a north and south pole, kind of like planet Earth does. So that's important because they have kind of a front end and a back end, and if they're going to move toward the light, they have to kind of coordinate their movement, all these individual cells in a colony. You might be asking yourself, if they show phototaxis, where is the organelle that has them detect light? And the answer is, it's in those individual flagellate cells of this green algae. I don't think that we will be able to see those structures with any of our microscopes, but if you have a really, really good one, then you can. All right, I'm going to pause this and show you a different magnification level. Okay, that was 40 times. Let's take a look at 100 magnification. And ooh, there we go. I think the one on the right is smiling at us. And now I'm going to zoom out, and let's see if we can get 400 and show you what that looks like. I have a separate clip where I'm going to show you the perimeter of one of these colonies so you can see the individual green cells, but you can sort of see them here. All right, so this is at a magnification of 400, which most of your scopes should be able to do. In fact, I think here you can see the 2 edges of these larger mother colonies, and if you see those little dots along the perimeter, each one of those is actually an individual Volvox cell. And if you're asking, I can't see those 2 flagella, well, you probably can't, because they're pretty transparent and difficult to see. But when you see the living ones, you will see the effects of their motion in the water, and that's kind of fun. All right, I'm going to stop this here and give you a separate clip showing you 1000 magnification.